Hello, and welcome to the Drug Discovery World podcast. My name is Giles, and I'm here to take you through another topical and insightful article from Drug Discovery World. Today's episode is taken from our winter 2017-18 issue, and is titled, Technology Innovation Will Help Prepare Drug Discovery for the Smart Screening Era. The article was written by Dr. Clive Green and Dr. Philip Spencer, both of AstraZeneca. So now on to the main article. Technology innovation will help prepare drug discovery for the smart screening era. Drug discovery is undergoing a transformation powered by advances that magnitude more molecules and enable smarter selection of compounds. We're on the cusp of having previously unimaginable amounts of information about each target and many more targets to prosecute. For instance, last year, AstraZeneca launched an integrated genomics initiative through which scientists will investigate data from as many as 2 million human genomes, including more than 500,000 from clinical trials run by the company. The possibility of being able to interrogate many more targets and learning more about the features that drive compound identification and optimization is a major step towards the ability to intelligently select compounds to screen in drug projects. The diversity screening commonly performed in pharma workflows today, those all-by-all screens, necessitated by our lack of knowledge about targets and compounds alike, could and should be replaced by smart screening of targets, with just the compounds deemed the most promising fits by strong scientific evidence. This will allow researchers to be more judicious about the use of samples, rather than wasting a proportion of each compound for every new screen, despite the low likelihood of most being effective. Smarter screening approaches would ease the pressure on compound libraries in pharma, biotech, and academia. Currently, the broad brush method of screening requires having large volumes of compound samples on hand at all times. These samples often have to be shuttled between lab facilities, and sometimes around the globe. Each diversity screen chips away at the volume remaining in the library, raising concerns that precious samples will be exhausted in routine screening and then unavailable when truly needed. Innovative automation can help alleviate this challenge and preserve compounds for the coming era of smart screening. At AstraZeneca, a collaboration with several vendors in compound storage and processing is allowing a rethink of compound library usage and approach. By pushing the boundaries for automation, labware, and software, they have made it possible to extract more value from each compound in readiness for an increase in new targets. This approach could serve as a template for other organizations looking to extend their own capabilities. They recognize the need for innovative automation solutions across many areas of drug discovery, and collaborations with academia and vendors will be an important element in delivering scientific impact from these investments. Opportunity. As drug discovery scientists generate new biological screening data about a host of targets, there is a pressing need to improve the management of compounds. Since no company has unlimited access to compounds, ensuring that the available samples last as long as possible is essential for safeguarding the success of future screens when new targets emerge. The challenge that faced the authors at AstraZeneca, one that will be familiar to any scientist involved in drug discovery, was increasing their capacity for high-throughput screening without depleting their compound library. AstraZeneca has more than 2 million compounds at its disposal, with approximately 150,000 at risk of depletion, and a cost of replacement of $50 to $100 per compound. At the same time, they continue to seek new collaborations with academic institutions and other external organizations. Each new project increases the number of high-throughput screening campaigns they would like to run. Addressing this challenge involved several separate technical goals. In order to achieve the aspiration of being able to select any compound for any screen, AstraZeneca needed more rapid automated storage and retrieval capabilities. Miniaturization was also necessary to make each compound last for many more screens. Ideally, scientists at the company's major research hubs would have local access to key compounds, rather than having to request them from a central facility and then have them shipped. That would require a different approach to store more samples in the same equipment footprint and conserve sample volumes across multiple locations. These were not trivial tasks. If they could be accomplished, though, it would result in workflows that would be much more amenable to the kind of smart screening AstraZeneca hoped to deploy. Technical approach. The authors at AstraZeneca recognised that a complex project like this could not be solved by a single technology platform or vendor. They approached vendors with whom they had established long-standing relationships for compound storage and retrieval, Brooks Life Sciences, liquid handling, LabSite, and IT systems, Titian Software, and addressed the challenge together. In addition to having the vendors work closely with AstraZeneca during this project, it was essential that they also worked in partnership so that any solutions would be integrated and fully automated, without the pitfalls of incompatible software or hardware, or requiring manual intervention to go from one platform to another. In one major step, AstraZeneca transitioned the liquid handling approach toward trays of tubes rather than traditional microplates. This required the liquid handling vendor to work hand in glove with their tube manufacturer, crafting specialized tubes compatible not only with the compound storage infrastructure, but also with the acoustic dispensing liquid handlers where they would ultimately be used. These innovations also led to a dramatic increase in the number of tubes that could be held in a tray, and a new barcoding system for meticulous sample tracking. Integrating software was also important. AstraZeneca's IT systems vendor teamed up with them and the other vendors to develop a software solution that could manage the entire process, including sample ordering, automated storage and retrieval, and the liquid handler protocols for screening. It also reports process results such as compound quality and location, plate barcodes, and inventory data. To break through what has been a technological barrier for acoustic liquid handling, AstraZeneca's liquid handling vendor developed a novel platform with innovative hardware and software features to support the transfer of samples from storage tubes. Additionally, it developed a complementary robotic platform to seamlessly integrate its acoustic liquid handler with tube and microplate handling devices in one modular system for a streamlined and contained process optimized for maximum throughput and efficiency. Technology development and validation for this completely novel approach has taken 2 years, and AstraZeneca will start to deploy this new solution this year. New capabilities. The improvements from this new integrated approach will be significant. According to models, the workflow will be able to support more than 70 high-throughput screening campaigns each year, a sizable increase from their previous approach. Automated storage and retrieval capacity will increase fourfold, from 20,000 to 80,000 samples per day, and using high-density trays that each hold 1,280 acoustic dispensing tubes will enable 100,000 samples to be plated in just 30 hours for a typical primary screening campaign. By creating a completely non-contact workflow using solely acoustic dispensing liquid handlers, rather than the current industry standard, a hybrid of contact and non-contact dispensing, the authors anticipate a 10-fold reduction in sample volume across all assays. The fully acoustic solution, with its groundbreaking acoustic tube design and innovative acoustic liquid handling technology, will streamline many processes and reduce the number of steps needed within compound management to generate plates for screening. For an organisation that runs as many assays as AstraZeneca, that represents significant cost savings. By increasing compound access at AstraZeneca's major research hubs, each site will hold the most relevant section of the company's library for its scientific requirements. Each site will have lower volumes of each sample, but because of the miniaturization made possible by acoustic dispensing technology, those samples will last much longer. The risk to AstraZeneca's 150,000 endangered compounds, for example, has been completely removed. Indeed, this shift would not have been possible without such a degree of miniaturization. There is simply not enough building space at some of AstraZeneca's sites to store as many as 1 million compounds in the volumes that would otherwise be needed for so many screens. Together, these advances will contribute to significant business impact. An extensive cost analysis determined that the total cost of ownership for this new system, with its dramatically higher throughput, is actually lower than the cost of the previous system at AstraZeneca. Other analyses have shown that the new approach will improve the overall quality of screening data generated, giving AstraZeneca's scientists a better foundation for their studies. Vendor collaborations. Despite the prevalence of laboratory technologies in drug discovery, there remain areas where innovation and investment in automation could offer real benefit to productivity and quality. In chemistry, for instance, there remains a predominance of manual handling activities, a situation that could be transformed with the kind of liquid handling automation and miniaturization implemented in the high-throughput screening process. At AstraZeneca, scientists are currently developing automated systems for many of the common chemistry tasks in the workflow of making, purifying, and analyzing compounds. And in screening, AstraZeneca's collaboration on acoustic mass spectrometry, where a mist of sample is ultrasonically ejected from a microplate well directly into a mass spectrometer, has demonstrated a significant reduction in assay development time and expense. At a speed of 3 samples per second, the use of acoustic MS in primary high-throughput screening reduces the percentage of false hits due to its label-free mass spectral data. These and other areas of opportunity can be addressed successfully with the sorts of multi-vendor collaboration described here for compound management. The kinds of sophisticated large-scale projects that pharma and biotech companies embark on often need so many resources and specific kinds of expertise that assembling a team of outside vendors is the only viable option for delivering a robust solution. Of course, it is also necessary to have realistic expectations about these large collaborations. Coordinating a project across several companies, all with individual objectives, inherently comes with a lot of risk. Mitigating this risk relies on the final products aligning with the strategic direction of all business units involved and a shared commitment among the teams. Throughout the collaborative process, all parties must continually evolve the plan and address risks. Also, a clear understanding of the total investment for such large, long-term projects is essential, as well as being able to put those numbers in the context of opportunity cost and the competitive advantages of the products. These aspects can be difficult to quantify, but they are critical factors in determining if a collaborative approach like this is worthwhile. Pooling resources. Throughout the drug discovery and development industry, more and more organizations have come to the realization that no individual company will be successful on its own. The need for collaborative efforts has led to an era of increased cooperation among pharma, biotech, and academic research centers. Compound sharing is one trend that has enhanced library diversity for a number of organizations. AstraZeneca, for instance, has teamed up with Sanofi to freely share nearly 250,000 compounds, and has an agreement with Bayer through which each pharma is able to screen new targets against both companies' entire compound portfolios. The trend to open up pharma compound collections for screening against academic research centre targets has gathered pace in recent years. At AstraZeneca, this has created the demand to support more than 30 additional hit-finding campaigns annually with subsets of the corporate compound collection. There are many such agreements in the industry today, underscoring a willingness to combine expertise in the interest of helping all organizations accelerate the time to finding a hit for any given target. One of the reasons these collaborations are so popular is that academic institutions are centers of fundamental biological understanding for the targets of interest in drug discovery. From generating more information about biological targets to screening compounds, projects that once consumed significant resources and risk at pharma and biotech companies are now being performed in academia, often funded by government or disease foundation grants. Many of these academic groups and their pharma partners are sharing novel compounds to help the broader research community. The European Lead Factory is an excellent example. 7 farmers pooled and donated 270,000 compounds, while smaller companies and academic teams contributed 230,000 compounds to create the joint European Compound Library. Similarly, EU OpenScreen is a new project that aims to use a share and share alike approach to encourage users of its compound library to contribute results from their own bioactivity research for the rest of the network. At AstraZeneca, scientists from the Medical Research Council and Cancer Research UK will share a collaborative space in the UK Centre for Lead Discovery at AstraZeneca's Research Centre in Cambridge, UK, accessing AstraZeneca's compound library to test novel drug targets. The progress being made from this spirit of collaboration will be a critical driver for developing effective new therapies. Distributing the challenges and costs of hit discovery across many more organisations, including academic research institutes, is a smart means of mitigating risk and accessing the best science. Looking ahead. Thanks to more frequent cooperative hit discovery programs, the need for increased screening capacity is pronounced. As we learn so much more about targets, drug discovery scientists must have access not only to more screening, but also to smarter screening. At AstraZeneca, this situation drove them to make a significant investment in rethinking their approach to compound screening. Everything from where those compounds are stored to how rapidly they can be accessed and assayed. With a multi-vendor collaboration, AstraZeneca have managed to develop and integrate new automation, labware, and software to enable a completely novel approach to compound storage and management, with major implications for screening capacity and speed, as well as reduced costs. While this will add real value to AstraZeneca's own discovery pipelines, it is an approach that could be implemented by any organisation willing to make such a commitment. Innovative automation and collaboration will be integral to a future of smart screening and accelerated drug discovery. This article was written by Dr. Clive Green and Dr. Philip Spencer. Dr. Clive Green is Executive Director and Head of Sample Management, Discovery Sciences, IMED Biotech Unit, AstraZeneca, Cambridge, UK, where he oversees the company's collection of human biological samples and small molecules. He spent more than 10 years as a medicinal chemist and earned his PhD in organic chemistry at the University of Nottingham. Dr. Philip Spencer is Team Leader of Sample Management, Discovery Sciences, IMED Biotech Unit, AstraZeneca, Cambridge, UK, where he is responsible for the global deployment of acoustic technologies. He has more than 15 years of experience working in compound management and earned his PhD in synthetic chemistry at the University of Sussex. If you've enjoyed this episode, then you can subscribe to the journal free of charge by visiting ddw-online.com, where you'll also be able to view full articles, including references, and download the original PDFs of past articles, including this one. If you've enjoyed the podcast, then as always, please do leave us a review and subscribe, and you can also follow us on LinkedIn and Twitter. Thanks for listening. We hope to see you in the next episode.